All right. Well, hello, and what an honor and privilege it is to bring the first in a series of national webinars to you. My name is Phi Mo, and I am the Arizona chapter president. And I hope that you and your family have been safe during this pandemic. I am grateful that the foundation is providing these webinars since we are not able to meet in person for our chapter meetings and gatherings. Let me share a little about our chapter and how we have engaged with our members and community to further the mission of the foundation. But first, I do want to give a shout out to Jackie and Dan Olson, who led our chapter for over 20 years. It is an honor to carry the legacy that they have created here in Arizona. Earlier this year, we held our 14th annual Vision Walk, led by our amazing chapter vice president, Patty Wolverton, where we had over 1,000 registered walkers. We are also proud to have one of the largest, if not the largest team in the nation, Team Ashley, led by Judy and Craig Hauger and their daughter, Ashley Rinder. We have raised over $1.1 million in walks and are proud to kick off the Vision Walk calendar season in late February. We have also started some new chapter-led fundraising and community awareness events in the past few years. Our Dining in the Dark dinners, which have been led by Coral Colbank, have given us an opportunity to showcase how the foundation's funded research has impacted us in Arizona. Last year, Dr. Christina Reichart from Tucson shared how her youngest daughter's life changed after receiving Luxturna, and how now she can see the blue in her sister's eyes and the smile on her parents' faces. We also give our guests, many who are fully sighted, the experience of dining with no or low vision. We have found our guests leave with a deeper appreciation and understanding of vision loss. We also organized our first golf tournament, led by Molly Thompson, which introduced the foundation to a new group of people. With the abundant amount of sunny days in Arizona, we hope to continue our golf fundraising events when it is safe to do so. Our engagement and awareness events include our virtual networking sessions, led by Nate Chrisman, our retinal practice outreach led by Patty Wolverton, local blind and visually impaired organizations outreach led by Doug McGuire, and diversity outreach led by Rhonda Carroll. I'm grateful for the dedication of our chapter members to continue the mission of the foundation. I'd now like to introduce Ben Shaberman, Senior Director of Scientific Outreach and Community Engagement for the Foundation Fighting Blindness. For 16 years, Ben has been reporting on retinal research for all of the foundation's electronic and print publications. In addition, he presents the latest scientific advancements in local and national events for patients and families, and conducts various training activities for staff and constituents. He enjoys working with constituents one-on-one to help them understand the retinal disease and the research underway that may benefit them. He's also a published author of a great sci-fi book, which I highly recommend, and a fellow running buddy. Welcome, Ben.
Well, thanks, Phi. I'm honored and privileged to not only present the latest in research for our chapters, but I'm honored to be introduced by you, Phi. I know you and the Arizona chapter do such a great job driving our mission. And it's also an honor to be what I call the opening act for Dr. Bernstein. I'm really looking forward to hearing about his latest news and information on nutrition for inherited retinal diseases. My overview will be pretty brief this time. It won't be a sweeping overview like I did earlier in the year for some of those webinars. I'm just going to cover a few projects that have been in the news or have had some interesting activity as of late. And we will have a Q&A session at the conclusion of Dr. Bernstein's presentation. And in the interim, you're free to submit your questions through the Q&A icon on your toolbar. And we will get to those again after Dr. Bernstein is done with his talk. So before I begin, I wanted to give a big thank you to our sponsors who make a lot of our outreach, including events like this, possible. Genentech is our visionary champion partner, and our outreach silver partners are AGTC, Elox Pharmaceuticals, Mira GTX, and Spark Therapeutics. So before I talk about some of the projects that have been in the news, I just wanted, as I usually do, to provide some context for where the research is from a clinical standpoint. And as I'm sure many of you know, we're at a very exciting juncture with somewhere in the neighborhood of 42 clinical trials underway for emerging therapies. And that number continues to increase steadily and, again, excitingly. Now, one thing I want you to keep in mind is while that number sounds very impressive, and it is impressive, there are two aspects to clinical trials that I think are important to understand. Number one is that not every therapy that's in a clinical trial is going to make it through and get regulatory approval, like from the FDA or the European Medicines Agency. And another thing about therapies in clinical trials is there are new technologies that aren't in clinical trials yet that are working to do things even better than those that are in clinical trials. So we have newer, for example, gene delivery technologies that are coming through the preclinical pipeline. And as many of you know, retinal diseases, the inherited retinal diseases are very diverse. Many genes are associated with them. And those are all important reasons why we need to keep the pipeline full. Yes, 42 clinical trials is a great milestone, but we need to keep bumping that number up. So the first project I want to cover is a drug, a molecule being developed by the company Nacuity out of Dallas. And the Foundation Fighting Blindness is investing $7.5 million in clinical development of that project. We also funded a lot of preclinical research for this molecule at Johns Hopkins over the last several years. And this drug is called NACA. And it's an oral therapy designed to reduce oxidative stress. Now, oxidative stress is something that our cells and our bodies go through as just a natural part of living. But when the cells or tissues are suffering, perhaps from a disease, oxidative stress increases. And researchers have found that it plays a significant role in exacerbating vision loss. It leads to increased cell death. So NACA is a molecule designed to mitigate oxidative stress to slow vision loss. And one of the nice aspects of this emerging drug is it works independent of the mutated gene causing the retinal disease. So regardless of what your gene is, there's a potential for this drug to work for you. Early on, the investigators are targeting RP and Usher syndrome, but it may apply to other conditions because oxidative stress is common to many inherited retinal diseases, or really all. So the great news that we've heard recently is that a clinical trial is underway in Australia for people with Usher syndrome. It's a phase 2 that was recently launched. If you're wondering why Australia, it's because that was the quickest way to get this into a trial given the COVID situation. And in Australia, they were able to identify a population of Usher syndrome folks who are ready to get involved in the clinical trial. Remember, Usher syndrome is RP with hearing loss, so you can really think about this as another form of RP. Then if this work goes well, a phase 2 for RP and Usher syndrome for NACA will be launched in the US in late 2021. So stay tuned to that. Now, another project that the foundation is funding very significantly is being developed by a company called Sparing Vision in France, a company that was spun out of the Institut de la Vision. And the foundation is funding 7 million euros. And we funded many years of lab research at the Institute to understand this particular therapy and help move it toward a clinical trial. And Sparing Vision just recently announced that they received an additional 44.5 million euros in venture capital investment, which is a nice boost to the clinical development. Now, those of you with retinitis pigmentosa, Usher syndrome, and some of the other related conditions know that your vision loss starts on the periphery. The initial symptoms of most of these conditions is night blindness and peripheral vision loss. And that's because your rods are affected first. Rods give us night vision and peripheral vision. But what the very smart researchers at the Institut de la Vision realized is that rods secrete a protein that helps keep cones alive. So as these diseases progress and people lose rods, cones are ultimately affected. And the researchers believe that it's the loss of rods that cause the cones to degenerate because this protein is not being made. So the researchers characterized this protein called rod-derived cone viability factor, or RDCVF, and they've developed a gene therapy that expresses this protein. So this too is designed to work independent of the mutated gene. And again, we're looking at it for outer retinal degenerations like RP, Usher syndrome, and potentially other conditions. And the hope is that by getting an injection, this protein will help preserve cones, which provide visual acuity, central vision, vision in lighted settings. Cones are really the cells that give us the most independence and the ability to do our activities of daily living. So Sparing Vision is getting ready for a clinical trial in 2021. They're in the process of setting up their production processes so they can be ready to launch a trial next year. So stay tuned for that. Now, another company that's been in the news recently is called Stargazer Pharmaceuticals. Again, some company that the foundation has funded. They're Boston-based. And I'll note that Stargazer, which is targeting people with Stargardt disease, is also using data and information from our natural history study for people with Stargardt disease called ProgStar. And they're using other consulting services from us to design and launch their clinical trial. So they too recently received additional funding in venture capital investment, $57 million. And so we're always very excited to see our investments leading to additional investments from other companies and other venture capital firms. That's one of our strategies. So to understand Stargardt disease, it's a condition that is caused by the accumulation of these toxins in the retina that cause macular degeneration. It's an inherited form of macular degeneration. Now, how those toxins are produced is through the processing of vitamin A. We all need vitamin A to see. Vitamin A is the magic molecule that makes our retinas light sensitive. But when vitamin A is metabolized in the retina, there are byproducts that are produced. And these byproducts, if they're not cleared properly, if they accumulate, cause problems, as they do in Stargardt disease. So the Stargazer drug is designed to reduce the uptake of vitamin A in the retina so that it slows and reduces the accumulation of these toxins. So excitingly, they've launched a phase 2A clinical trial. They completed their phase 1 in healthy subjects to make sure it was safe. And hopefully we'll see some positive results from the phase 2A trial. So the last project I want to feature is being developed by a company called Videre Bio, and it's an optogenetic therapy, and I'll explain what that means in just a moment. But what's exciting is that Videre Bio was recently acquired by the big pharmaceutical company Novartis for $280 million. Now, we funded Videre to get their clinical development going, and we're very excited that Novartis has acquired the company for two reasons. It puts a big financial foundation behind development of the drug. And as an investor in Videre Bio, we actually get returns from the Novartis acquisition. And what do we do with those returns? We invest them back into research. So again, our investments are not only helping pave the way for therapies like Videre, they're helping to boost other research investments as well. So let me explain what optogenetics is, and I'm going to refer to this little diagram on the right-hand corner of my slide. It's a side view of the retina. And toward the top of this diagram, you see those long cells, including the cone-shaped cells that are red, green, and blue, and then the straighter cells, which are the rod cells. Those are photoreceptors. Those are the cells that make vision possible. Now I have this ominous X going through the photoreceptors to signify somebody who's lost all their vision to a retinal disease like RP or age-related macular degeneration. A lot of therapies in the pipeline aren't going to help people who have lost all their photoreceptors. A gene therapy that delivers a good gene to replace the bad gene is not going to work because there are no photoreceptors left to receive it. But some very ingenious researchers saw that another layer of cells below the photoreceptors in this diagram, called ganglion cells, survive after photoreceptors are lost. And I have them circled. And what they did is they, in their lab studies, they delivered a gene to these ganglion cells that makes them light sensitive and work something like photoreceptors. So it's using ganglion cells for vision. Now, this is a gene therapy, but it's designed to work independent of the mutated gene or the disease causing vision loss. Now, the work by Videre came out of the lab of John Flannery and Ehud Isikoff at UC Berkeley. And what's exciting about their approach is they're using a gene therapy that expresses a protein that's very similar to the protein that makes, or one of the proteins that makes our photoreceptors light sensitive. Earlier this year, or in previous talks, you may have heard me or other researchers talk about other optogenetic strategies. There are actually 3 clinical trials underway for other optogenetic strategies. But those strategies are using a protein that's not like the normal, natural human protein. And in fact, the other proteins will only work in extremely bright light. So the protein, the opsin, as we call it, that Flannery and Isikoff have developed should enable people, if it works, to see in more natural settings of varying light. Again, this is for restoring vision in people with advanced disease, and it does work independent of the gene. We're not at a clinical trial yet for this, but stay tuned. Again, the Novartis investment really boosts the move toward a clinical trial. So to close out, I just want to give you your ongoing assignment as people who and families who have retinal diseases. It's really important that you see your eye care professional on a regular basis, even though they may not be able to deliver a treatment or even get you into a clinical trial right now. They can help manage complications like cystoid macular edema. They perhaps can address a cataract. There are other conditions of the eye that you need to watch out for, something like glaucoma or other conditions. And perhaps the eye care professional can update your eyeglasses prescription if that's a needed thing. So see your eye doctor on a regular basis. Next, it's really important for you to be genetically tested to identify the mutated gene that's causing your vision loss. Now, there are more than 270 genes, each of which, when mutated, can cause a retinal disease. But once you identify that mutated gene, you get a clearer diagnosis, and you can get on the radar screen of investigators conducting clinical trials. Many of the trials that are underway will require you to know your mutated gene. And we have an excellent no-cost genetic testing program. More information about that is on our website, fightingblindness.org. You can read all about it. And it's ultimately your doctor who orders the test online through our partner, Blueprint Genetics. And our program also provides excellent genetic counseling at no cost through Informed DNA to help you understand the results. And so also, whether or not you know your mutated gene, it's important to be in My Retina Tracker. It's at myretinatracker.org. This is our patient registry. It protects your privacy. It's highly secure. But researchers and companies conducting clinical trials are using My Retina Tracker for recruitment. And I know many of you out there are very involved with the foundation in many different ways. We really appreciate that. And it's through you that we can continue to drive our sight-saving mission. So thank you. Again, visit fightingblindness.org. If you want to learn about clinical trials, visit clinicaltrials.gov or our website. There's a lot of information on our website about the ongoing trials. And we strongly encourage you to get involved in My Retina Tracker at myretinatracker.org. So again, after Dr. Bernstein's presentation, we'll take questions. So I'm going to stop sharing my screen. And next up, I believe, is Tim, correct?
That is correct. Thank you, Ben. Thank you. That was great information as always. We really appreciate all you do for the foundation and for everybody who's benefiting from all the research. So thank you. And welcome everyone. Hello. And I am thrilled and honored to be able to introduce our speaker for today. And before I do that, I want to give you all a little bit of an idea of what our Cincinnati, Northern Kentucky chapter is all about. The Cincinnati and Northern Kentucky as a community was actually one of the participants in the first Vision Walk in 2005, a couple years prior to us even becoming a chapter. Our walks have raised just shy of $1.3 million in 15 years. We've had 6,029 walkers and an average of 29 teams per year. Our largest walk was 760 walkers in 2014. At the time, one of the largest number of attendees for any walk. And at one time, we had the record for the largest walk team of 130 members, which I think has now been eclipsed by Arizona. So thank you for that, Phi. we have to get that back at some point. Our oldest walker, Mary Wolpert, was 92 when she walked with us in 2009, and as far as I know, that she might be to this day the oldest ever to join in a walk. Our walk is also the closest walk to Delta Gamma's national headquarters, just up the road in Columbus. So we have held the record for the most DG teams at a walk. That's 7. It's comprised of active houses and alumni groups. Fundraising efforts that funnel into our vision walk have included a wine and beer tastings, golf tournaments, silent auctions, more wine and beer tastings. Cincinnati loves its beer. We've had beer sales at the world's 2nd largest Oktoberfest here, and even homemade pickles, among other things. We also have the best media chair. And I have to say that because it's my wife who's the main anchor of the CBS affiliate here in Cincinnati. We began hosting meetings in 2005, before we were even officially a chapter again. Presentations have featured speakers from greater Cincinnati area, as well as from the FFB, addressing advances in research and treatments for IRDs, orientation and mobility, and nutrition, among other topics. In 2018, we had 65 in attendance as we hosted a local participant in the Luxturno trials who told us of the first time she saw stars at the age of 18. We've hosted coffee social support group meetings and panel discussions about the importance of the relationships we have with our caregivers, and Save Site Saturday events with many regional services represented addressing accessibility, technology, orientation and mobility, and other services. Several years ago, we gained representation within the Greater Cincinnati Vision Coalition, which also includes Cincinnati Association for the Blind and Visually Impaired. And that's an organization we partner with quite frequently. There's Prevent Blindness, Cincinnati Children's, Clovernick Center for the Blind, Cincinnati Eye Institute's CEI Foundation, among others. And the coalition's work is intended to improve the delivery of eye care and vision-related services through meaningful collaboration to leverage our collective resources to better serve the greater Cincinnati community. I'm very grateful for our volunteers and our gracious and selfless chapter leadership team. Included in that is Jim Shirk, my vice president, Peggy Mitchell, in charge of outreach, Susan Farber, Dan Dufresne, Doug Tiger, Liz Lyons, Bryson Rahindran. And special thanks to Robin Chadwick, who really kickstarted our Vision Walk and our chapter in 2005 when she started the Vision Walk for her son, Zach. I joined them in introducing Dr. Bernstein. And as a reminder, again, as Ben said, we will be addressing questions during the Q&A portion of the webinar. I now would like to introduce Paul Bernstein, MD, PhD, Val A. and Edith D. Green Presidential Professor of Ophthalmology and Visual Sciences at Moran Eye Center, University of Utah. Dr. Bernstein joined the faculty of the Moran Eye Center of the University of Utah in 1995, where he currently divides his time equally between clinical and basic science retina research and a clinical practice devoted to medical and surgical treatment of diseases of the retina and vitreous, with special emphasis on macular and retinal degenerations. He was promoted to Professor of Ophthalmology and Visual Sciences in 2007 and awarded an endowed professorship in 2008. Dr. Bernstein's current research and interest focus on biochemistry and biophysics of nutritional interventions against inherited and acquired ocular disorders. Please join me in welcoming Dr. Paul Bernstein. Dr. Bernstein.
Thank you very much for allowing me to be one of the first webinar leaders of this, one of the first webinar speakers. And I will share my screen here. Well, thank you very much. I have a long history with the Foundation Fighting Blindness. They've supported my work for many, many years on a number of interesting projects through the years. I'll talk some about them today. And I've been on the scientific advisory board of FFB for quite a long time to help make sure that they have very good science to support the interventions against inherited retina diseases. So this is an outline of my talk, and I'm gonna be talking about nutrition in the eye. And some people may wanna know, why am I so interested in nutrition? Where does that come from? Well, it comes from the fact that I have a long history of a hobby in botany and horticulture, actually. I've been very interested in trees. And in fact, I'm a tree collector, which is a fairly unique kind of hobby. And I'm showing here, these are stock photos. These are not actual trees of mine, but of some of the rarest trees in my collection, both the Chihuahua spruce and the Wollemi pine. The Chihuahua spruce was discovered less than 100 years ago in a remote grove in northern Mexico. I have one growing outdoors. It's thriving very well here, and it's one of the only ones in the United States. I also have a Wollemi pine, which is even rarer, was only recently discovered in Australia in 1993, and it's a living fossil that was thought to be gone along many years ago. And it's, again, doing very well, but that has to grow in a greenhouse. So I've been interested in plants and natural products through the years, dating back all the way to my PhD work, where I worked on vitamin A metabolism in the eye. And I'm going to be talking today about various natural products that are important in inherited retina diseases and in age-related macular degeneration. And it will be divided into two different parts. I'll be talking about specific interventions. These are mechanism-based, and the non-specific interventions that are more broadly based. And Ben has given a little bit of a good background of antioxidants, and we'll talk some about that later on in the talk today. And I look forward to giving a lot of to fielding your questions that you'll have at the end, because there is so much to be covered in this very interesting topic. So why should we be considering to intervene nutritionally against inherited retina diseases? Well, through the years, as we understand the biochemistry, the underlying causes of some inherited retina diseases logically point to nutritional interventions, that they are mechanism-based. It's very logical. This is something we should be doing in many of these diseases. Also, being natural products, they often have fewer side effects than some of the pharmacological interventions. These are products that nature has developed. Obviously, nature does have some poisons and some toxins out there, but nature really does give us, by and large, gives us very on balance, gives us things that are very beneficial for us. And so they tend to have fewer side effects, and because of that, patient compliance can be excellent. They're often, but not always, relatively low cost. And there's truly strong patient and clinician interest that I've seen through the years in such interventions, because it really empowers many of us in a way to try to go against some of these inherited retina diseases. So one of the diseases that very early on was known to be a nutritional disease is a very rare disease. It's known as gyrate atrophy. This is so rare that I will say that I've not actually seen a patient with this. It's estimated that less than 1 in 1 million people have this condition worldwide. But for various reasons, it's more common in Finland, 1 in 50,000. Patients who have this get an interesting form of, let me get the laser pointer here. Let me get pointer options. They get a very characteristic degeneration of the peripheral retina that's very scalloped out here. And with relative preservation of the center of the vision. This condition is so characteristic that it shows up on many board questions of ophthalmologists. We're supposed to be able to recognize this just by seeing it. And it also is a syndromic disease. The patients also get cataracts, muscle wasting, hair thinning, and various brain abnormalities also. And it was learned many years ago, before we even understood a lot about genetics, that this was a defect in ornithine metabolism, which is a type of amino acid. And ornithine is not an essential amino acid, but it is something that is important in formation of various amino acids, including arginine, glutamic acid, ones that are essential and are very important in protein synthesis. And if you have a defect in ornithine aminotransferase, it then causes problems and buildup of toxins, including ornithine can be very high, and that's toxic to various tissues in the body and to the retina. And so this is treated with a low protein, low arginine diet and vitamin B6. And so any patients that are identified with this, and we would pick this up now on genetic testing, should be treated with this sort of diet. Another one that is very rare is a disease called Refsum disease. And this is found in less than 1 in 250,000. And it's a mutation in the PHYX gene, or the PEX7 gene. And this is, again, another syndrome that it gives rather characteristic findings of retinitis pigmentosa with bone spicules in the periphery. But they also get cataracts, get ichthyosis, which is skin problems, deafness, neuropathy, and limb and digit abnormalities. And this has to do with the fact that there's an inability to metabolize an unusual fatty acid called phytanic acid. It's a branched chain fatty acid. And it's treated, again, by altering the diet, not by taking a supplement, but you have a diet that's low in phytol and phytanic acid. And that means it's a fairly strict diet for these patients. They have no green leafy vegetables, no animal fats, and no milk products. So essentially, they're just eating cereal products and various, and so it's a relatively unpleasant diet, I would think, to be on. So, and then even going more rare is a disease called A-beta lipoproteinemia. This is an ultra rare disease, retinitis pigmentosa, and less than 100 people have been described worldwide, and this is mostly in Ashkenazi Jews. And this is known as Bassen-Kornzweig disease. And this is a defect in a protein that disrupts metabolism of fat-soluble vitamins. And specifically, the very important one that happens is that there's a deficiency of vitamin A effectively in these patients. And they develop an eye disease, they develop a retinal degeneration, they develop abnormal electroretinograms. And this is treated with, once we identify this disease, is treated with high-dose vitamin A and vitamin E. And this was a very important finding, not only because, and suggested that we should be considering treating retinitis pigmentosa back, in the 1990s when Bernstein was working on his clinical studies, that it might be important in treating other forms of retinitis pigmentosa. And we'll talk about that a little later in this talk. And finally, the next one is visual cycle defects. We know that that vitamin A is very, very important. It's one of the key molecules of the of vision. And vitamin A is found bound in its active form, which is known as 11-cis retinal, is bound to the protein rhodopsin in all of our rod cells. And this is the pigment that's required for detecting light. Vitamin A then is, after it absorbs light, then goes from its 11-cis form to the all-trans form. This is the more common form that we would see in our diet, but this is not visually active for us. The vitamin A has to go into the retinal pigment epithelium cells, and there are enzymes there, including LRAT, lecithin retinal acyltransferase, and RPE65, that make this go back into its active form, the 11-cis form, and that goes back into the photoreceptors so that we can detect light. Defects in some of these enzymes, specifically RPE65, which is what Luxturna treats, can disrupt this visual cycle, so we no longer can detect light and the visual cycle does not function properly. And this is a cause of retinitis pigmentosa and Leber's congenital amaurosis. And we've heard that the very exciting work of genetics to try to genetically fix RPE65, and yes, this does work. But the thought is gene therapy is an invasive, expensive treatment. Would there be possibilities to just supplement vitamin A and get this cycle to work better? The problem is if we put in all trans and you still have defective enzymes in this process, it's not going to make the 11 cis. So you naturally might think, well, could we just give 11 cis retinal or 11 cis retinol and charge up the visual cycle? And the answer is it doesn't work. 11 cis is too unstable. It breaks down very quickly in the body down to the all trans form. But some researchers have realized that we could give a different form called the 9 cis form, which does work as a visual cycle chromophore. It can bind to opsin to make isorhodopsin, and this can charge up the visual cycle. So there have been small research trials that have been conducted, giving various forms of 9 cis to people. And yes, indeed, their vision does seem to improve. These studies are still small, and we still need to understand the toxicity because these are vitamin A compounds, both as the 9 cis acetate form shown here, or as a 9 cis carotenoid that it would be cleaved to the 9 cis form. But this is very exciting alternative way of pharmacologically treating various inherited retina diseases that where the visual cycle is the site of the problem. And shown here is a little bit of data from studies, one by Dr. Scholl et al., where they were able to show that there was improvement in patients that were taking some of these 9 cis compounds using 9 cis retinal acetate. And another one of my associates that I know quite well, Dr. Rotenstreich, has used the 9 cis form of beta carotene that he derives from various algae and has used that. And so these are other compounds that are in trials and may be coming someday. Another nutritionally important based disease, in part, is Stargardt 1 disease. This is the common form of Stargardt disease that is inherited in a recessive manner. It's one of the more common inherited retina diseases that we see that affects the macula. And this is caused by defects in a gene called ABCA4. ABCA4 is very important for moving vitamin A from after bleaching in the visual cycle, moving it out of the photoreceptors and helping it move on to the retinal pigment epithelium. If there is a problem in this gene, in this protein caused by a genetic defect, we start accumulating toxic forms of vitamin A. The vitamin A is very reactive, the all-trans retinal, and it starts reacting with some of the fats in the discs shown right here. And then it starts forming various toxic compounds called bis-retinoids. And typically these are 2 vitamin A molecules that are stuck onto various other compounds here. And these are very difficult to metabolize, so they get stuck in the tissues in the retinal pigment epithelium, and they can cause light-mediated damage. They can also cause oxidative stress damage. So we think that it's bad to be forming these bis-retinoids in the eye, and so we need to, because these are nutritional compounds, are based on nutritional compounds on vitamin A, we've been trying to look at, from a basic science standpoint, how to alter this. Now, Ben has talked about one of the studies, I think it's the Stargazer study, where they are essentially causing a mild to moderate vitamin A deficiency by disrupting the pathways of vitamin A within the body by altering the retinal binding protein. So that is one approach. Another approach is shown here, is to make a special form of vitamin A where the hydrogens at a certain point are substituted with a deuterium, a heavy form of hydrogen. This can still work just fine in the visual cycle and does all of the good things that we want it to do, such as charging opsin and making rhodopsin. But having these 3 deuteriums here disrupts the formation of the bad compounds, the A2E. And shown here, this is a compound made by a company known by Alkeus in animals, and also they think in humans as well, that when we replace much of the vitamin A in the diet using this deuterated vitamin A, there's less formation of A2E, and the hope is that this will decrease the rate of formation of the toxic compounds and slow the rate of Stargardt disease. The studies are in progress. We participated at the Moran Eye Center in some of these studies, but the results have still not been released. So we're still waiting and hoping that this compound could work. I've talked about the common form of Stargardt disease, but there are other forms that look just like Stargardt disease and share the same name. And one of them is called autosomal dominant Stargardt disease, or Stargardt 3. This, as I said, looks a lot like Stargardt disease, but it has a completely different gene and a completely different mechanism. And it causes about less than 5% of Stargardt disease, and it's due to a completely different gene and different protein. And the gene is called ELOVL4. And this was discovered by one of my colleagues, Dr. Kang Zhang. And at almost the same time, we found another family in Utah with a similar defect, with a comparable defect in the ELOVL4 gene. And both of these mutations lead to disruption of this protein. And this protein, we learned, is very, very important in making fatty acids in the retina. And they are shown here, and that what they do is they make a very unusual form of fatty acids that are very long, much longer than the ones we normally eat in our diet. The fatty acids we think are important for the retina are the ones known as long-chain fatty acids, docosahexaenoic acid and icosapentaenoic acid. These are EPA and DHA, which we get primarily from seafood. These are important in making the fats that keep our photoreceptors happy. But our photoreceptors need even longer ones, the ones that are up to 38 carbons long. And even though we don't eat them in our diet, we need to have them in our retina. And we have, fortunately, for normal eyes, we have the gene ELOVL4, the protein, that makes these longer and longer ones. This then allows us to have normal functioning of our retina. If you have a defect in ELOVL4, you don't make these, and your photoreceptors begin to dysfunction, and you get a Stargardt-like condition. When we found this, we thought, well, since this is a dominant disease, could we try to give even more of the precursors, the EPA and the arachidonic acid, possibly the DHA, to make these very long-chain fatty acids? And is it possible that my patients I had 18 patients in one family with this, and we noticed that some of the patients had very mild disease, actually had almost normal retinas going into their age 50 and 60, and some of them were legally blind in their 20s. What was the difference? Well, we were able to find that nutrition and diet was very important in this case, because the people who ate fish were the ones who had the normal retinas, while the ones who didn't eat fish at all, and there are a lot of people like this in Utah because we're a landlocked state, that were doing very poorly. So we were able to show in an epidemiology study that this diet was mediating changes in this disease. And we even tried working with this family to get them to eat more fish, to eat more, to take fish oil. And it's hard to change habits when you're an adult, but they are at least working with their children as they identify them. and are encouraging them to eat fish. And they seem to be doing better, but this is a small family, so it's not easy to prove in a clinical trial. The even more exciting thing is that we know that these sorts of defects in ELO-VL4 function also happen in age-related macular degeneration, so it's much more generalizable. And you could ask, well, why not, even if you can't eat a diet that's high in retinas and testicles and other things that are high in long-chain fatty acids, very long-chain fatty acids, couldn't we just make them and give them to people? Well, it turns out that it's very hard to make these, and for years, chemists couldn't make enough to even give to a mouse. So I recruited some of the best chemists at the University of Utah, and we now can make gram quantities of some of these very long-chain fatty acids. And thanks to the support of the Foundation Fighting Blindness, we are testing this right now in mice. We can make pure compounds, and we find that in mice, when we administer them, we can see it going in the bloodstream within hours, and we can see its delivery into the retina within just a matter of 8 to 24 hours. And even more exciting, we've been able to see in these mice that they do see even better, and we're starting to correct defects in mice with ELO-VL4 defects. So this is how we develop new nutritional supplements for this eye. This one we know occurs in nature. We don't normally consume it, but we're very excited that if we can continue doing these moving the basic science to eventually making enough to give to humans, that we may have an impact on inherited retina disease and even age-related macular degeneration. And finally, for the other nutrients that are specific, I'll talk about a disease that until recently we didn't even know was an inherited retina disease. That's something called macular telangiectasia type 2, or MACTEL. This is a disease that we thought was sporadic. We didn't really know that it was inherited, but I was part of a research project sponsored by the Lowy Medical Research Institute, where we looked at large Utah families, and we saw indeed that this disease, this macular disease that occurs typically in the 40s and 50s and knocks out reading vision, and that we could see that it was indeed inherited in a complex manner. We could tell there were multiple genes going on, and that it had late onset and what we call variable penetrance. It didn't always cause problems in some people's eyes, probably based on their nutrition. We were able to find from a family where a father and son and other members in the family had this, and also a neurologic disease, that we were able to find the first gene called serine palmityltransferase, or SPT. And we were able to see that instead of the normal pathway where serine is combined with palmitate to make some compounds known as sphinganine and ceramides that are important for retinal and neural health, that the enzyme starts using alanine, an incorrect substrate, and makes all sorts of toxic compounds. So this is a new disease that we now know, a new inherited disease. We know some of the mechanisms that serine and alanine is involved. And this suggests another nutritional intervention that we can do, which is to give high dose serine. We already know this works for the neurologic formations of the disease. And we in the Lowy Medical Research Institute are instituting a trial to look at very high doses, 30 grams a day of serine, to see if that can help. Now I'm going to turn to non-specific interventions in the last 10 minutes or so. And this has to do with the fact that not all problems in the eye are related to specific biochemistry defects. We know that retinitis pigmentosa now is caused by hundreds of different genes, and that's why we need to find approaches that could be more generalizable, so that each project, is not, each gene is a major multi-million-dollar project to get that to work. So are there simpler nutritional compounds that we could use? So the first of these was done by one of my mentors from many years ago, Dr. Elliot Bernstein, who had understood the importance of vitamin A in the eye and the fact that there were diseases like Bass and Cornswag syndrome that where vitamin A deficient, relative vitamin A deficiency is a problem. So he conducted trials of giving high dose vitamin A, 15,000 international units a day, in a randomized placebo-controlled trial on hundreds and hundreds of RP patients. And he was able to show, to his credit, that there was a slowing in progression of the disease over many years when people took this vitamin A supplement. So this was an important early study of the 1990s, but it was controversial from the start. And this had to do with the fact that although this was statistically significant, there was a lot of questions whether this was clinically significant, whether the patients really would notice a change in vision, because they were using their outcome being an ERG amplitude, not actual functional vision back then. There was also worries about systemic and ocular toxicity, as well as the fact that it is a teratogenic compound. It can cause birth defects if giving it at high levels. So this was the first step. It was a proof of principle, and it had early impact on the disease. But I would say that it's not I still have some patients who are taking high dose vitamin A, but I would say the vast majority of my patients have opted to things that are a little safer and a little more biochemically based. One of the things that we think is interesting and still working on are some of the other fatty acids. I already talked about the very long chain fatty acids, but the long chain fatty acids are much more common in our diet. They are fundamental components of making our photoreceptor discs here, and they help keep your rhodopsin happy in the discs here. They're commonly found in fish oil. And so again, Dr. Bernstein did some studies to see if supplementing with DHA and EPA could help in the health of the retina. It turns out that in retinitis pigmentosa, they were not able to prove that it really made a difference. These can be very difficult studies because many of us eat fish in our diet. It's very hard to control that in a large clinical study. Other people, like David Birch, have looked at supplementation for X-linked retinitis pigmentosa. Again, there were signals that it could be interesting, but so far these have not proven to be effective. And so many patients do take these still as being eye healthy. And we're hoping, as I've said before, about some of these very long chain fatty acids, that those might be even more important in future supplements. And shown here is just, again, some of the research that Dr. Bernstein was showing changes in electroretinograms, both shown by David Birch's group and Bernstein's group. So there were a few subtle signals, but it was not enough to really to change clinical practice. Then the next one I'll talk about are carotenoids. And this is something I've been studying for most of my academic career at the Moran Eye Center, in fact, all of it. And these are very, very interesting compounds. These are lutein, zeaxanthin, and mesozeaxanthin. And I was attracted to these because the eye naturally goes out of its way to concentrate these compounds that we get from dark green leafy vegetables right in the macula, and shown as a cross-section of a monkey retina. And for those of you who can see color very well, and you can see that the macula itself is yellow in color. In fact, the macula is the macula lutea, or the yellow spot, because these compounds are naturally concentrated in the eye. We think this is very important, or maybe very important, in macular degeneration. And I'll talk about a little more of the data in a few slides ahead. And we were able to show, but we also thought it might be important in preserving the macula and preserving where the cones are. And so we developed methods to measure the amounts of lutein and zeaxanthin in retinitis pigmentosa and choroideremia patients to see if this could be important. And it turned out that levels were perfectly normal in retinitis pigmentosa and choroideremia patients. But in Stargardt patients, they're very low, which suggests that it might be useful in trying to replenish it in the Stargardt patients. Bernstein did try to do clinical trials with lutein supplements, and they did not show any benefit. So it's not there's no specific recommendation for lutein and zeaxanthin for RP. Why should these be important for eye disease? Well, they are very efficient antioxidants that are concentrated in the retina. They're also yellow compounds, so they screen the hazardous blue light that we're all exposed to every day. They're very stable in the retina, and they're very, very highly concentrated. In fact, they're the most concentrated anywhere in the body there. Where do we get luteins from? Well, we get them from dietary sources. We get them from dark green leafy vegetables, and we consume about 1 or 2 milligrams per day. They're very safe. You can consume up to 75 milligrams per day, according to the European Food Safety Administration. And most supplements are using 6 to 10 milligrams per day. And we get them both from plant sources and from eggs or other good sources. Zeaxanthin's a little harder to come by. You have to consume orange and yellow fruits and vegetables, very specific ones, things like yellow corn, orange peppers, or Japanese persimmons. We in the US consume about 0.2 milligrams per day, but the acceptable dose is considered up to about 50 milligrams per day. And most trials are using somewhere between 2 and 6 milligrams per day. Egg yolk is also a very good bioavailable source of this. A third carotenoid in our macula is something called mesozeaxanthin. That's not normally in our diet. Unless you eat a lot of shrimp shells and fish scales, but it's made in the retina by the enzyme, actually RPE65 enzyme, and its natural precursor is lutein to make mesozeaxanthin. So we don't consume it in our diet, but supplements are available and marketed in the United States. So there are 2 options for that, either supplements or diet. And they screen blue light, they're antioxidants, and they also seem to prevent formation of A2E. And they're concentrated from an early age. In fact, when you're born, you already have lutein and zeaxanthin concentrated in the macula of your eye. They enhance, so they also have a functional effect. They improve our contrast sensitivity. They decrease glare, so we can tolerate glare better. And they even subtly improve visual acuity. A number of studies have shown a couple letter increase. This may not be clinically significant, but it is statistically significant. And I've shown here, just to summarize, some of the RP studies. Bernstein did not find any protection giving lutein and zeaxanthin. And we found that in patients with retinitis pigmentosa and choroideremia, that they had similar amounts in their retinas. But it is very important in age-related macular degeneration. We know that age-related macular degeneration is the most common cause of vision loss in the elderly, and it is in part an inherited disease. We have non-modifiable risk factors like age and heredity are very important, but we also have many modifiable risk factors such as smoking, cardiovascular disease, alcohol consumption, light exposure, but especially nutrition. And since this is such a common disease and so it uses so much of our healthcare resources, we'd like to prevent the disease if possible. So through epidemiology, we have found that a number of antioxidant, vitamins, minerals, and other nutrients seem to be protective. And specifically, one of the ones that comes up very, very commonly are the carotenoids, lutein and zeaxanthin, from epidemiology studies. And one of my mentors, Joanna Seddon, published in 1994 that patients who consume large amounts of lutein and zeaxanthin in their diet and who have high blood levels have almost a 50% lower risk of developing advanced age-related macular degeneration. And based on this information, the National Eye Institute conducted the Age-Related Eye Disease Study 2. And this was to try to improve the original antioxidant compounds of the formulation of AREDs and make it safer, that they enrolled 4,000 patients and gave them either an original AREDS 1 supplement or a modified one that would have 10 milligrams of lutein and 2 milligrams of zeaxanthin. This is about 5 to 10 times as much as we normally consume in our diet. And they also tested omega-3 fatty acids and followed them for 5 years to look at progression to advanced age-related macular degeneration. I was one of the investigators and enrolled about 60 of these 4,000 subjects. And in 2013, we were able to show that we could improve on this formula, and that adding lutein and zeaxanthin did improve the efficacy of this formula modestly from 30%, from decreasing the progression to 34% down to 30% within 5 years. And it also was safer and was associated with lower risk of cancer and other problems associated with high doses of beta carotene. So we now have a modified, the new formula of AREDS2, which we recommend to all patients who have moderate to advanced age-related macular degeneration in one eye at least, and who are at risk of progression to advanced in their fellow eye. And this is shown here. We still recommend vitamin C at 500 milligrams per day, vitamin E at 400 international units, lutein at 10 milligrams per day, and zeaxanthin at 2 milligrams per day. And we also have zinc oxide at either 25 or 80 milligrams per day, and copper at 2 milligrams per day. And this is standard of care that we recommend for all patients at high risk for progression to advanced AMD. And so we still, for AMD, I still recommend, especially for the worried well, the younger people who are bringing their elderly family members in for treatment for AMD, that diet is still the most important thing. To eat a healthy diet with lots of fruits and vegetables, to eat fish, and to have no excessive fat, and that they should take a supplement if they're in the high risk range. There's still many supplements to come and many herbals, but those we have to wait until there's more data. And of course, we recommend that they don't smoke, and we think that they should avoid excessive light exposure, and that they should participate in future clinical studies. And we're hoping that some of these very long chain fatty acids might be a good substitute for the less successful omega-3 fatty acids from fish oil. And then I won't go into detail on this because Ben did a great job. There are antioxidant supplements that we're looking at, and N-acetylcysteine, the NACA one that he was talking about, is something that's being tested right now. Other antioxidants haven't fared as well, such as vitamin E did not turn out to be very efficacious, very successful for treating retinitis pigmentosa, and actually might have had some toxic effects. But we're very excited. I'm going to be part of the NACA study, and we're very excited about its future potential. And then many inherited retina disease patients ask if they should be taking AREDS and AREDS2 supplements, and the answer is we still don't know. And I still always learn from my patients. And one of the things that I learn from my patients are about herbal compounds. And so there isn't a lot of good science to really prove that any of the compounds shown here, such as bilberry, anthocyanidins, whether they could slow down retina disease. Eyebright has a great name, but we're still not sure why it's really healthy for the eye. There's some studies that suggest that red wine and resveratrol in it may be very helpful against inherited retina disease, and studies are in progress. And of course, I'm always humbled by the fact that goji berries, which is from Chinese traditional medicine, is something that has been known for centuries to be thought to be helpful for the eyes. And this is actually one of the best sources of zeaxanthin. So that shows that herbal supplementation may be helpful. And then, of course, remember, more is not always better. There can be compounds that can be toxic to the eye and crystallize in the eye. Canthaxanthin, shown right here, is a skin tanning agent that crystallizes in the eye. Niacin, which some people take to lower cholesterol, can cause cystoid macular edema. Oxalic acid, which we can get in spinach and green leafy vegetables, can also have toxic effects on the eye. And even lutein and zeaxanthin in rare situations in some of my patients can crystallize and cause problems in the eye. So just remember, everything in moderation, be wise in what you use in nutritional supplements. So in conclusion, Mechanism-based nutritional interventions against inherited retinal diseases play a definite role in a small array of rare diseases that we discussed. Newer mechanistic approaches are in early stage clinical trials: deuterated vitamin A, 9-cis retinoids, 9-cis carotenoids, serine, and less specific nutritional approaches might help, but the clinical data is still hard to come by, but we are doing our best to really try to put good science behind retinoids, omega-3 fatty acids, very long chain fatty acids, carotenoids, antioxidants, and herbals. And of course, I want to thank the various people who have kept my laboratory going: the National Eye Institute, the Foundation Fighting Blindness, the Lowy Medical Research Institute, and Research to Prevent Blindness, and of course, the people who are members of my laboratory. So thank you very much, and I'm happy to answer any questions.
Great. Thank you, Dr. Bernstein. That was outstanding.
Okay, I will unshare.
Okay, so a lot of questions. And one thing I want to let people know, we have about 15, 20 minutes left. If we don't get to your question, we'll follow up. So you will get an answer to your question, even if you don't get it today. But a general question that somewhat encapsulates a few questions I got: what are your general dietary recommendations for an RP patient?
Okay. All right. So I have this talk many, many times with my patients here in Utah. People are interested in nutrition and diet by quite a bit. So I do talk, I start with the green leafy vegetables and the carotenoid rich compounds. I think that the science is very, very good for that. We have the basic science. We know that these are healthy compounds for heart disease and everything else. So I encourage a diet: dark green leafy vegetables, orange and yellow fruits and vegetables. I think that fish and the omega-3 fatty acids are something that could be, that I encourage. It's interesting that the epidemiology has consistently shown that people who consume fish do very well with regard to eye disease. Our purified supplements have not fared as well, which means, it tells me that we're missing something in the fish and the fish oils. For those of people who are strict vegetarians and don't want to consume fish, there are algae versions that also are available on the market, and I encourage them. With regard to, there are other, for age-related macular degeneration, other inherited retina diseases, we think that a healthy body weight, having a normal BMI, is important. Getting exercise. These are all good things that you're going to do, and they're good things for the eye. Only as I showed in the toxicity, you have to be taking a lot of lutein. You have to be taking probably 80, 100 milligrams a day. You're taking many, many times what we would give in the supplement to start running into problems. So some people ask, well, should I be taking supplements? And the answer is, we don't know yet. We haven't been able to do a lot of studies in the worried well, people who may have genetic predisposition to macular degeneration. We're hoping to do those trials. They're difficult to do because they take many, many years to do.
Thank you. That was great. In terms of fish, it sounds like you do recommend that people consume fish.
Yes.
Is there a type of fish that you specifically recommend?
Yes. So what we think is the oilier the better. So that's why we say cold. When we say cold water fish have a lot of these omega-3 fatty acids. So we're talking about salmon, tuna, those sorts of things are very, very good. Mackerel, people are not quite as fond of, but fish oil is what we're looking for. So what is a fish that's probably not as good? I guess you're talking about things like catfish, things that may not be quite as good. I'm not sure about tilapia, some of the ones that are the warmer water fish, but cold water fish is the way to go. That's what we think.
Great, thank you. So those are the things that you should eat. What are the foods or supplements you should not take. And we got a question about vitamin E. I know Dr. Bernstein commented on that. And we also got questions about caffeine and alcohol.
Mhm.
So curious
Okay.
what you think.
So that's a good question. We think that we take a lot of cues, I think, from the heart disease people. And so in terms of your fats, we think that polyunsaturated fats and especially the omega-3s are the ones that are less inflammatory and are probably better for the eyes as well. We don't know this for sure. And some of the very long chain fatty acids, we're going to look at some of the omega-6s, but for right now, you want the omega-3s, and that tends to be more the fish oils. The omega-6s and the trans fatty acids and some of the other ones that we think are the bad fats for cardiovascular disease are ones that are probably not good for the eye, we think would be the same. So, and then saturated fats, again, are probably not helpful. With regard to monounsaturated fats, things like olive oil, there's less proof for those in the eye. But on the other hand, there is epidemiology. We talk about the Mediterranean diet. That has been very consistent, high in olive oil, and is something that might be useful for the eye. So those are the things that I would look at. In terms of foods to avoid, I'd say mostly the fats. Now, we know smoking is bad. With regard to caffeine, I haven't seen anything strong either way. I think that, so I don't advise for or against caffeine.
Yay.
Okay. Do what you want. And it's interesting in epidemiology, there's some that goes back and forth all the time on caffeine, whether it's good or bad.
Right.
So I will, I wrote an article recently on whether dark chocolate would be helpful for the eyes, and the answer is, I said it's go for the taste, not for what you're doing for the eyes. The effects were so subtle on that. And with regard to alcohol, most studies have shown that alcohol in moderation, we're talking about 1 or 2 glasses of wine a day or a beer or 2 a day, is not bad for the eyes and may possibly be good. So alcohol in moderation. Obviously, high doses of alcohol and all the nutritional problems that go with that are not good. And so that's what I would recommend.
Great. Nice overview. So a question about your study on that very long chain polyunsaturated fatty acid that you don't get from your diet that's produced in the retina. So you're studying that in the dominant Stargardt disease model. Do you think if it shows some promise in that model, it may apply to other models or other diseases like RP or AMD?
Yes, we are very excited about this. We have some interesting data already on this, that it could be useful. I would say age-related macular degeneration we're quite interested in. We have found, we're looking into whether it could be used in other parts of even the lifespan to retinopathy of prematurity. And for inherited retina diseases, we think that we see these abnormalities in a number of different models. So we think that this is an exciting new approach to this. And so we're pushing us forward on this, and we have our chemists on board to be making more and more of these compounds and to try it. So, and
Great.
we're really impressed. One of the things that I found most interesting is when we give this to mice, it goes to the retina, and it goes specifically to the retina. It doesn't go to the liver. It doesn't go to other parts. It doesn't go to the brain. The retina really wants these compounds. And so stay tuned. We hope that we'll find something more with this.
Interesting. Well, we're excited to fund it, and hopefully we'll get some good results. A question about saffron and turmeric. What do you think of those as possible supplements?
So those I classify as going into the herbals, and we have looked at them. I mean, obviously the Indian and Asian literature has seen some important, some interesting effects, and they're very widely available. I have done a little bit of work, but I would say the doses that we have to give are pretty high, and I I think that it could be interesting. I don't know if I don't have enough good evidence to recommend it on a scientific basis yet.
Okay. So I want to get back to vitamin A and Stargardt disease. This continually comes up, and you did mention it in your presentation, but I want to repeat that vitamin A supplementation for Stargardt disease patients is not advised because we're concerned it could exacerbate the disease. What kind of diet do you recommend for a Stargardt disease patient? Is it okay for them to eat fruits and vegetables, including those that might be somewhat higher in vitamin A?
Sure. So one of the things that you need to understand, or that we all need to understand, is that there are various different forms of what's called vitamin A in our diet. And many times when you buy a supplement at your health food store, they say vitamin A, but you have to look at what really is the form of vitamin A. The form that really raises your levels very quickly are vitamin A palmitate, vitamin A acetate. These are the preformed vitamin A. We don't eat a lot of preformed vitamin A in our diet. And the best source of preformed vitamin A is liver. And specifically the story that we always hear in medical school is polar bear liver is your best source of really high vitamin A, enough to make you toxic. So I would say for Stargardt patients, yeah, just avoid liver is probably a good idea. Now, the more common way that we get vitamin A is beta carotene and various other carotenoids in our diet. Now, lutein and zeaxanthin actually don't have any vitamin A activity at all. They can't be made into vitamin A. But beta carotene that we get from cantaloupe, oranges, all the things like that, the beta carotene and carrots that we get is made to vitamin A, but we have enzymes that convert it and make only the amount of vitamin A that we need. So a supplement that has beta carotene in it is probably pretty safe. But so I think that I tell patients basically a normal fruit and vegetable diet is probably going to be just fine. Liver, stay away from. I would stay away from preformed vitamin A supplements, the palmitate and the acetate. And if you're going to take a beta carotene type supplement, a couple thousand units a day is probably reasonable. That's very similar to what we're getting in our diet. I wouldn't go specifically out for a beta carotene supplement if you have Stargardt disease or something related.
Great, thank you. And again, that's for Stargardt disease, just to
Yes.
remind people.
Yeah, Stargardt disease. Yeah, it's a very specific one, and in terms of its vitamin A, the A2E doesn't build up in a lot of other diseases, so you don't have to worry about that. But Stargardt disease, it really does, and that's why, with the Stargazer, they're trying to cause a vitamin A deficiency, a moderate vitamin A deficiency. So that tells you you're going the wrong way if the Stargardt disease, if you're taking a vitamin A supplement.
Right. Good point. Another question, not nutrition related, unless you think of bright light as being part of your diet, but it's a question we get a lot. And it's sort of a two-part question. What do you think most people should do in just their daily living in terms of bright light exposure? And could bright light exposure during an office visit with an eye doctor, if that eye doctor isn't aware of our diseases, very up on our world, could that be problematic?
Okay. So light toxicity is a concern. We know that excessive bright light can damage your retina. That's very well known. But we're talking about the studies that we know well, especially for age-related macular degeneration, are based on the extremes. These are fishermen, these are people who are farmers out all day, every day in the bright sunlight. And yes, if they wore hats and sunglasses, they had lower rates of age-related macular degeneration. For inherited retina diseases like RP, there are some, we think that the retina is already has oxidative stress and needs, and the light could be exacerbating that. So yes, it's a good idea as long as it's not inhibiting your daily activities to be wearing sunglasses and a hat on a bright sunny day. With regard to the lights that many of us are exposed to indoors, the blue light from our office lights, from our computers, that's controversial. People are trying. I don't think it's been proven either way, so I'm not gonna. The blue light can also cause problems with circadian rhythms. Whether it causes degeneration is not proven, but avoiding excess is good. With regard to, I think the last question, when you see your eye doctor, yes, we do use some pretty bright lights when we're looking at patients. I still use it, I have to say, because it gives me what I think is valuable information. We often turn down the blue light when we're doing things like autofluorescence in Stargardt patients and minimize the amount, but I don't think you should avoid seeing your doctor. We will do your best for you to try to minimize the exposure and just make it a few brief seconds if possible.
Great. Well, thank you, Dr. Bernstein. We're getting close to 1:30 here on the East Coast. So we're going to wrap it up with a final comment. Again, Dr. Bernstein, thank you so much for a wonderful presentation and taking time out of your busy week to be with us. But we're going to close out with some final comments from the director of our chapter program, Renee Paulsell. So Renee, I am going to bring up my final slide and give you the mic.
All right, thank you so much, everyone, for this wonderful presentation. Thank you so much for all of these questions. I know we've probably got a number left in the queue, and we will get back to you and make sure that we have those available as we get these questions answered. So again, thank you so much for being with us today. We are delighted to present these programs. We will have another one coming up in January, and it will be on gene therapy, and you will be getting information on that. Please go to our website, fightingblindness.org, and those will be listed and you can register, and we're delighted to bring those to you. One final note, I did want to, both Tim and Phi, I wanted to thank them for being with us and the graciousness of their chapters for hosting our presentation today. Thank you, gentlemen, and we greatly appreciate the work that you've done. I also want to thank all of you across the country who participated in all of our walks. This past October, we raised over $1 million in support of the Foundation Fighting Blindness, and we're so grateful for that. We do want to make sure that everyone is aware of our spring vision walks. Our spring vision walks will be held virtually again this year, simply too challenging with COVID-19 to bring folks together, and you will be hearing about that in your local communities as we move forward with our June date. And last, I want to remind you that this is our 50th anniversary coming up in 2021, and we're delighted to begin our celebration of the 50th anniversary of the Foundation Fighting Blindness with our Hope from Home Gala. That will be held on National Rare Disease Day, which is February 28th. This will be an absolutely remarkable evening with lots and lots of special, wonderful fun for everyone. And we will be sending out some emails. If you've not gotten one, please check your inbox. Please check your spam folder to make sure you've gotten that. And if you don't have it, you again can visit our website and get that information as well. A gala right from your home that you can celebrate all of the great things that the Foundation Fighting Blindness are doing, in addition to really recognizing National Rare Disease Day. Again, we greatly appreciate your time being with us today. This has been a remarkable Saturday. And again, we look forward to presenting these to you every other month, beginning in January with our next session on the third Saturday in January, talking about gene therapy. Again, for all of us at the Foundation Fighting Blindness, have a wonderful Thanksgiving, and we appreciate you being with us today. Thank you.
Happy Thanksgiving, everyone.
Thank you, you too, Ben. Thank you, everyone.